Hello everyone. I guess I'll just start, and if anyone else wants to join, they can. Um, I will be discussing whatever's happened in the past week, week 3. The I'll give a sort of an brief of week 3, and in case you have any doubts, you can ask me. Okay. And I would like to request everyone to mute them if unless they have questions. Okay. Uh Let me just share the screen. Okay. Can everyone see the presentation? Okay. Thank you. The this week, uh, Sir has talked about briefly about lipids and carbohydrates, mostly focused on genetics. Uh, this is a sort of a review of last week as well, wherein we saw that a protein after translation exists in an unfolded state, and immediately in a very short span of time it folds and goes into its native or folded state, which is more stable. The energy difference between the unfolded and native state is around 5 to 10 kilocals. um, and this is basically the thermodynamics associated with it. uh, the factor that contributes most to how the protein is folded and what structure it can form uh, is mostly water. Like if the protein has too many amino acids which are hydrophobic in nature, uh, because their R group is hydrophobic, they would tend to come together and try to minimize the surface area that is in contact with the water. uh, there are also electrostatic interactions such as salt bridges and hydrogen bonds and dipole-dipole interactions that would uh, play a role in how the protein is shaped and what structure it takes up, what tertiary structure it takes up. Disulfide bridges also exist, but water has a little less role to play, and more the composition of the protein plays a role in how where the disulfide bridges are formed. uh, there would be metal binding sites, and these pockets are designed such that they can hold a metal ion, and the protein sort of folds around such that this pocket is present. And if you have mutations, there might be a reduction or increase in the entropy of the protein. Um So when you talk about the stability of the protein between the, as I said, between the unfolded and the folded or native state, there is a difference between of 5 to 10 kilocals, which even though is very small, when you do the calculations, you realize that the equilibrium is sort of tilted more towards the folded protein side, wherein if for every one molecule of unfolded protein, you have to the 2 into 10 to the power 7 molecules of folded protein, which is a lot. Uh, Now, this 5 to 10 kilocalories per mole uh, of energy is a very small number if you consider all of the energy equations that come into play while the folding begins. Uh, So there are three major problems associated with this. The first one is that uh, the protein, if you can recollect the Ramachandran's plot, one there can be multiple uh, directions the protein can fold at, not many but multiple directions. So every protein has to sample all of those directions to fold itself before it reaches its native state, and this folding would take a lot of time if it were to sample uh, all of these positions. And this it would have to do for all the amino acids that's present in the whole stretch. So this creates this paradox that it requires a lot of time to for a protein to fold itself to its correct state. And there is also the second point being that the there is a linear relationship between the sequence, the primary structure, and the final folded tertiary structure. The one particular sequence of amino acids will always result in that particular folded structure, and this almost always happens unless your protein is misfolded, but those are due to other reasons. So it's given that it has to sample so many structures, it has to uh, account for all the heterogeneity that's present in the cell. It How is it that the relationship between the sequence and the final folded state always remains the same? The third point being that uh, even though there is a drop in the energy in when you talk about the native state, the the stability isn't that much. And um if you consider all of the equations, the drop should be more. So these are the three problems that are associated, that are sort of open questions associated with the protein folding that are still being studied. uh, this is one of the classic experiments to study classical experiments that were used to discover the relationship between the linear sequence of amino acids and its unique folded state. Uh, what uh, what they found was that ribonuclease A, which is the enzyme that is responsible for degrading RNA, is uh, so that enzyme in its native state has disulfide bridges, multiple disulfide bridges. So they what they did was that basically they denatured the protein and such that they have a linear stretch of amino acids, and they saw if on upon inversion the structure that it forms is the same structure that it had formed earlier. So first they treated the protein with reducing agent, which basically cleaves off all the uh, disulfide bridges, and then heated it. So heating basically results in disruption of the hydrogen bonds, van der Waal interactions, everything, and uh, it basically gives you a linear stretch of DNA. And then after denaturation, they renatured the protein by either cooling the tube or by removing the urea. And what they saw was that even though the disulfide bridges do not exist, because if you want to re create the disulfide bridges, you need some other extra proteins which are called chaperones, which were not present in this setup. But even though these disulfide bridges were absent, they saw that the structure that the protein had um, it had when only the disulfide bridges were disrupted was still intact. So um, the the thermodynamics of the protein folding is sort of like a funnel in which you have a linear unfolded state, and it sort of keeps decreasing its energy until it has reached the most stable state. And the, as I said before, the water molecules that surround that protein has a great role to play in it. So if suppose you have this stretch which is super hydrophobic, then what would happen is that this portion would try to fold in uh, itself such that it has very less surface area exposed to outside, as you can see here. So uh, the more the surface area, the more number of water molecules that can interact with that piece of protein. uh, so if it's hydrophobic, it will basically essentially try to reduce the number of interactions with water, and this is what happens, and there is a drop in entropy in that case. uh, and these are all of the major forces that come into play while we are considering protein folding: hydrophobic effect, hydrogen bonds, van der Waal interactions. But the drop in this energy is sort of matched, almost matched by the chain conformational entropy, wherein you what your structure is, that also has an entropy. And this results in your net stability of the protein. This is just an at the depiction that says the same thing, that a protein from its unfolded state goes through many different each part of it goes through many different stages to reach the most stable state. Now coming to enzymes. So enzymes are these uh, molecules uh, that I don't, okay, there is no graph, but I can draw. So when you are talking about a, um chemical reaction, what happens is that, suppose, sorry for the bad drawing, but yeah, if this is your reactant state and this is your product state, then what happens is that your reactant is here and suppose your product is here. To reach from here to here, your reaction uh needs an act. So this is what is called as an activation energy. It's supposed to kickstart the reaction so that it reaches a transition state. So the position here is a transition state, and then it uh, further drops its energy and gets to the product form. So what the enzymes do is that they have certain qualities, that such as some enzymes have these pockets in which they can sequester uh, the reactant molecules and then change their conformation, and this would lead to a faster production of the product, so to say. uh, if there were no enzymes, what ideally should happen is that all the reactant molecules will keep colliding, and if they collide in the right conformation at the right time, only then they will achieve the activation energy and then form the product. So what enzymes do is they basically reduce the activation energy, and this also results in a faster rate of reaction. um, we have been talking a lot about water and how it's important in protein folding and all that. That's because 70% of the cell is water by weight. The and This impacts hydrogen bonding and what and This would result in how your lipids, how your proteins, and how your DNA, everything is structured. Uh, Now, coming to lipids. Lipids are one of the lip, lipids basically form most of the all of the membranes in the cell, well, whether it is cell membrane or nuclear membrane, mitochondrial membrane, everything. If you talk about membrane, there is a lipid bilayer associated with it, and it forms it. I say lipid bilayer because if this is the outside and this is the inside of the cell, the so in a lipid there is the hydrophilic head, because because it can interact with water, and a hydrophobic tail. This hydrophobic tail is always towards the inside of the lipid bilayer. So this is what is called a bilayer, because there are two layers of lipids, and the hydrophilic head uh, is always outside of the cell. uh, there is this like the protein data bank, there is this database for lipids as well, known as maps, where you can see what the structures are of the fatty acids. So every so this hydrophilic head is from a glycerol, uh, is a modified glycerol, so to say, and this is a fatty acid chain. um can Yeah. So the major the so there exist different types of fatty acids as well, which have one tail, which have two tail. The In most cases, the fatty acids present in the cell membrane are that have two fatty acid tails. uh, if if there were one tail, it would be very difficult for them to be stable enough to form a bilayer. Rather, they'll form a micelle like structure, like you see in soap bubbles. So depending on what your fatty acid chain is and what your polar head is, there are various different fatty acids. And so these are different types of fatty acids, and these are the ones that are present most in the cell membranes. There are and uh, Apart from them being responsible for maintaining the membrane, they are also responsible for how for various communications that happen between the cells or inside the cell. Apart from proteins, lipids, nucleic acid, there are also carbohydrates that are present in the cell. And this happens to be one of the most abundant thing in the world. Uh, this when you talk about carbohydrates, there are depending upon how big your carbohydrate is, there are monosaccharides, which are just one. Uh, so suppose this is a monosaccharide, because it just has one group at one glucose group in it. There can be disaccharides such as lactose, which have a glucose and a galactose. Or there can be multiple there can be multiple glucose, fructose, galactose molecules associated, and they form a chain. If the chain is between 7 to 9 uh, uh, monosaccharides, then it's called something as oligosaccharides. There is If you think about big carbohydrates such as starch, they are called as polysaccharides, because the number of unit carbohydrates is very large in these. Um, Apart from them being the energy units, or them being associated in various energy cycle in the cell, they are also present in a large number at the surface of the cell. So the surface of the cell is decorated by the cell membrane just does not have lipids on it, it also has various proteins. These proteins and these lipids on the surface are also associated with different kinds of uh, carbohydrate moieties. Uh, and what so you can it's like a big brush border that you can see on the cell surface when you try to focus on the cell surface. uh, and how these carbohydrates are arranged in the cell surface dictates how they can migrate, how they can function in the body. Um these are some of the these are some of the carbohydrates that are associated. So these are uh, associated with proteins, the heparan sulfate, chondroitin sulfate, etc. There are also so this is a lipid, this is a chain of carbohydrates that's associated with lipids. And your protein, as I said before, your proteins, suppose this is your cell surface, your proteins and lipids both will have various carbohydrates conjugated to them. That's it. Going on to the next. Now changing tracks. We'll be talking about genetics, in particular Mendelian genetics. So when we say Mendelian genetics, it's mostly associated with um, the classical experiments of Gregor Mendel on pea plants. So yeah, when you talk about Mendelian genetics, you study the postulates that Mendel had proposed based on the experiments that he did on the pea plant. Uh, now, some of these postulates hold true, and some of these rules are not they have been modified, or they have been proved to be incorrect. So, when you talk about the human genome, uh, it has uh, 22 autosomes and one pair of sex chromosomes. So, when and each chromosome has multiple is arranged in form of chromatid, and each chromosome has multiple genes inside it. So, and every gene can be is stored in pairs inside the nucleus of the cell. So it has two forms. These two forms can be different, and these different forms are called as alleles in case of genes. And uh, there are the size of the 22 chromosomes and X and Y chromosome is not the same. The size depends on the base pairs that is present inside the inside each chromosome. And yeah, so this is one chromosome. While replication, what happens is that this chromosome sort of duplicates and forms sister chromatids, as you can see here. And these sister chromatids can then become disjoint, and they separate out and go into the daughter cells. um so if this is suppose chromosome 1 in your if this is the chromosome 1 in pea plant, say, um each there are two homologous chromosomes encoding the information that is supposed to be present in chromosome 1. They can be identical, but they can also have differences. Now, coming to the model that Mendel used to propose his postulates, he used pea plant, which and he used seven characteristics to propose the postulates, which were seed coat or color, which was gray or purple depending upon the flower color. uh, he also used seed color, which can be yellow and green, seed shape, which can be smooth or wrinkled, pod color, green or yellow, pod shape, inflated or pinched, uh, or smooth and wrinkled. You can also call it smooth and wrinkled. Stem height, tall or short, and flower position, axial or terminal. Um, so there is Mendel was lucky in the sense that none of these characters that he chose to study had any linkages associated with it. So when linkage means that if you say have two genes in two genes that you are trying to study, there might be chances, if they are present on the same chromosome, that these two genes will always be inherited together. So you won't find uh, the and the more distant these genes are spatially, the lesser probability they have to be inherited together. um so what he did during his experiments is that so the pea plant has seven chromosomes in it. What he did is that to and pea plant is heteromorphic in nature. So what he did is that he removed the stamens of the pea plant and bagged the bag the flower so that it cannot self pollinate or cross pollinate by itself. It can be pollinated in a very controlled fashion by manually brushing pollen from another plant or of interest. uh, and then you study different characteristics of that plant. The advantages associated with this are, because these are diploid plants, you get more variation. They undergo sexual reproduction and not asexual, so you can control uh, the reproduction, and there are a large number of seeds that you get from each cross, and you can screen these seeds and you get to know what the phenotype and genotype was. They Some experiments can be done by self fertilizing the flower, but most of the experiments, if you want to control it, you need to do cross fertilization. Now, coming to Mendel's postulates, what he postulated was that there exist unit factors in pairs uh, inside the genome of the pea plant, which control what the phenotype of the uh, plant would be. Uh, in other words, these unifactors were genes. Uh, and he said that the unifactors don't mix or contaminate each other. This is one of the assumptions associated with this postulate. Um, he also postulated that it associated with every gene there is a uh, in every gene there is a factor which is dominant and there is a factor which is recessive, and this is complete. Like if say x is dominant, capital X is dominant, it has to be dominant throughout. And if small x is dominant, recessive, it has to be recessive throughout. This postulate has been modified because there are there have been studies which have showed incomplete dominance and complete and co-dominance such, and I'll be discussing these later. um, So basically, dominance means that the effect of the dominant allele will show up, whereas the recessive allele will be repressed. Um, Segregation is the third postulate, in which the genes would segregate randomly, and there is no association in how the genes would segregate, which has been proved incorrect by the linkage studies done by Morgan. The it He also postulated that the genes would like the alleles would assort independently, like if uh, sorry I the independent assortment was disproved by linkage. And segregation basically means that the alleles would segregate randomly. Yeah, so these were the four uh, postulates that Mendel has had proposed based on his studies in the pea plant, which took a lot of time to come into the limelight be, and to be studied. Uh, and later on, people found out that these unit factors and are essentially genes in homologous chromosomes. So this is the structure of a chromosome, in which this is the centromere and these are the two arms of the chromosome. Um, and each chromosome, since these are the two homologous chromosomes, these each chromosome can have uh, different, slightly different modified versions of the gene. And coming to the monohybrid process, which in which you only study one particular trait. In this case, the P shape is studied, whether the seeds are wrinkled or smooth. Um if there are also dihybrid and trihybrid process, in which you consider two or three traits, or tetrahybrid process, as far as you want to go. um Coming to the terminology, the first generation from which you start is called the parental or P generation. The cross that you generate from the parental generation is the F1 generation, and when you self cross the F1 generation, you get F2 generation, and with every cross, you go down a generation. There also exist reciprocal crosses, in which you determine if the trait depends upon the sex of the organism, if the trait that you are studying is linked to females or it's linked to males. If you want to do that, what you do is, if you have two traits, say A and uh, uh, B, which you have two phenotypes A and B, you track down their genotype, and then use suppose, taking this example, female smooth seeds and male wrinkle seeds, and female wrinkle seeds cross male smooth seeds, you basically invert the sex of the parents and see if the outcome changes. uh, this is what's known as a Punnett square, which you use when you are trying to show the result of your cross. So this is the P1 generation, the parent generation, which it's a P generation from which you get gametes. So since this is a dominant uh, uh, this is a dominant phenotype and a homozygous phenotype, the gamete that you get is only T, capital T. And since this is a recessive phenotype and genotype, you get small t, which cross and give you the F1 generation or first filial generation, capital T, small t. these plants will be tall. So this was tall, this was dwarf, but these would be tall. Then you self cross the plants that are in F1 generation, like and so since it's TT, what the gametes you would get is either going to be capital T or small t. And so across between uh, a capital T sperm and a small t and a capital T egg would give you a tall plant, and even the heterozygous case would yield in a tall plant. Only case in which you get a dwarf plant is when you cross a small T sperm with a small T egg. So what you get is a phenotypic ratio of three tall and one dwarf. But when you consider the genotype, it's one TT to 1:2:1 is the genotypic ratio. Okay, moving on. Uh, so there exists a lot of in the past there existed a lot of debate in what the whether to follow the Mendelian genetics or the Darwinian genetics. So when you talk about Mendelian genetics, you follow Mendel's postulates that there is one allele that completely dominates the other. All of the alleles have two forms, and they're monogenic. They don't have any linkages. They assort independently, etc. Coming to Darwin, what Darwin found was that uh, if you have heard about the Darwin finches, is that he proposed that there is already a variation that exists in a population, a phenotypic variation, which uh, which and this variation can be passed on to the next generation of progenies, and the individuals which have better traits survive better, and uh, basically it's the survival of the fittest philosophy that Darwin genetics follows. uh, a lot of After a lot of experiments, what we have now is that a combination of Darwin selection and Mendelian inheritance. But to do this, you need to study populations of individuals, and which will show you how much genetic variation is exists within a population. And yeah um, Now, coming to non Mendelian genetics, the rules of Mendel only hold for pea plants uh, for those specific traits associated in the pea plants. If you considered some other trait which had some linkage, you would not get that result. So, the new genetics terms that have arose in after Mendel is incomplete. One of them is incomplete dominance, in which you have, suppose uh, you had, okay, sorry, some trait small t and some trait capital T. Your capital T would result in a tall plant. Okay. Your small t would result in a dwarf plant. But your capital T, small t would not result in a tall plant if there were a case of incomplete dominance. Rather, it would result in a plant which is of intermediate height. Like both the alleles come in and play a role in deciding what the uh, uh, height of the plant is going to be. Similarly, codominance, in which suppose you have uh, two if you consider coat color of an animal, say, you have a color which is brown in color, and you have a coat color which is white in color. What if you had a brown, you would it would result in a brown colored coat. If you had a WW, it would result in a white color coat. But if you had a brown and a B and a W, you would result in a mixed coat, a coat which is both brown and white, which is essentially codominance. uh, Penetration is associated with the gene, uh, and how much the gene can express itself. Suppose you had a gene which is say 100% active, like which generates 100% 100 proteins in one organism and 50 proteins in the other. You'd say the gene has 100% penetrance in the first organism and 50% in the second. This is explaining this very in a very lame in very layman terms. There are much more details associated with it which you can read upon. uh, Next, sex linked inheritance, in which you find that suppose so suppose there are two. In humans there are two sex chromosomes, X and Y. So there are certain genes associated with these two chromosomes. Suppose you have the suppose you consider the disease hemophilia. So hemophilia is known to be a X-linked uh, uh, disease, X-linked recessive disease. So it would be more common in males, because males only have one copy of the males only have one X in them. This is since they and this would uh, differ uh, between both the sexes of an organism. Therefore, sex-linked inheritance. There also exists dosage compensation, which has been catching up in the past few years, wherein so females essentially have two X chromosomes, right? But males have one X and one Y. So, say you have a gene A in the X chromosome, males would only have some 10 levels in the uh, of the gene A. But females, if dosage compensation wasn't present, they'll have 10 plus 10 from the other. So what dosage compensation means that is that the genes present on one of the X's is not expressed, it's silent. So that the level of the protein A is same in both the genders. Um, Coming to epigenetics in I yeah coming to epigenetics, there is even though you have a particular allele, if you are talking in terms of alleles, if even though you have a particular set of allele or a gene associated with an organism A, and the same allele set with B, the expression of the protein might not be the same in both the organisms, because of epigenetic factors, of because of how the uh, the gene the because of how the epigenetic factors function over that gene. It also happens in the human body, in which you have, say, cell A and cell B. Since it's in the same human, the both the cells should ideally have the same genotypic signature associated with them, but cell A can make hemoglobin, whereas cell B cannot. because the epigenetic factors associated with cell A ensure that the hemoglobin gene is in the euchromatic region of the cell, of the nucleus, whereas it's in the heterochromatic region in the, or it's not, it's silenced in B, and this results in a differential expression of genes within the same organism. uh, there can be multiple alleles that affect the same traits. Some of the examples are the eye color of humans, or the hair color or of humans, skin color of humans. In these conditions, multiple alleles affect how what the phenotype of that individual is. There are also lethal alleles which play a role in how play a role in the mm, in shaping the population of an organism. There might be some alleles which say you have capital T and small t, coming going back to that uh, example. Uh, Simplistically speaking, capital T would get more sunlight, whereas small t wouldn't. So capital T would, in a bush, in a tropical area where the trees are very high, capital T can survive because it has more sunlight, it has access to sunlight, whereas the small t does not. So an individual which has small t would not be able to access sunlight and therefore won't live. This is this is explaining this in very simple terms. When you talk about lethal alleles, it also uh, matters where, suppose you are talking about development of a baby. There are, of any animal, there are certain genes which are so lethal that they are prenatal in nature, which means that if they are not expressed at a certain stage of their development, the baby would essentially die. And there are certain genes which are, if not expressed in the later halves of the individual's lifetime, it would die. And this essentially means lethal alleles. um, Epistasis essentially means that suppose you have two genes, gene A and gene B. When you talk about epistasis, there is an effect of gene A over gene, how gene B would express itself. Suppose if gene A were recessive in nature, such as the uh, phenotype is small a, and gene B was like this, it would express the result that you'd expect from gene B would happen. Whereas when you have something such as a heterozygous condition of gene A and even a homozygously dominant condition of gene B, you would see that the result that you would have gotten from the gene B would not happen, because gene A is somehow affecting the expression of gene. This is epistasis. Pleiotropy means that uh, so earlier notion was that one gene, one effect. Pleiotropy essentially means one gene, many effect. The gene that is responsible for tallness in pea plants can also be responsible for the flower size, say in pea plants. I'm not sure about expressivity, and uh, yeah, other terms you can read up on, I guess. So basically, read up on imprinting, expressivity. These are not very, all of these are not very simple uh, concepts. They are very complex, and it, it would be a good exercise to go and read about these, what their genotypes can be, etc. Even when if you are considering epistasis itself is like a 40 minutes, can be a 40 minutes lecture from an expert easily. So it's a better idea to go read up on it, and in case you have any doubts, you can ask me in the next class. Now coming to different types of, whether the direction of genetics, so to say. um, when you say vertical genetics, you it's a gene flow between, suppose these are the parents and this is the progeny. It's the if When you talk about vertical genetics, it's basically the inflow of genetic information from the parents to the progeny, which usually happens when the gametes fuse. Uh, there can also be sorry, one second. There can also be inflow of genetic information from, say, organism A to organism B. Uh, Say you have two bacterial species, or bacteria A and bacteria B. Horizontal gene transfers are very common in bacteria. Uh, what would happen is that a piece of DNA from bacteria A, would flow into a bacteria B and integrate itself in the genome of bacteria B, and then express itself. So this is essentially called as horizontal gene transfer. The the inflow of information in vertical genetics is not only, so if you have a father and you have a mother, because of of, in case of humans, because how the gametes are, the ovum is very big, right? The ovum has all the material that the um, zygote needs until it's implanted in the uterine lining uh, of the mom, so to say. So until that point, it needs some RNA, some protein for to sustain it until it implants. So the mother would also give some extra things to the progeny, in terms of mitochondria and in terms of the RNA. So this is called as maternal RNA. And until implantation, until the zygote can make its RNA itself, which is called as maternal to zygotic transition, um, until it can make its RNA itself, it uses the maternal RNA and can therefore survive. The the mitochondrial RNA is usually comes from the mother. However, there are more studies on it ongoing, but yeah, mitochondria is inherently thought to be maternally inherited. So all the genetics that is associated with mitochondria would be the same as that present in the mother. um, the new genetics, so to say, after modern synthesis, which was after Mendelian genetics, uh, talks about neutral theory, in which you don't any the uh, environment does not play any effect, or the negative selection, in which certain components of the environment or behavior of that organism affect its survivability in a negative manner. There has been a lot of studies that show how horizontal gene transfer happens, and how it, even in cross species manner, like within a plant and a bacteria, etc., epigenetics, evolution, and uh, all of these sort of combine to form the postmodern synthesis. Now, coming back to Mendelian, coming back to modern synthesis, so to say, um, there are different types of mutations that might happen in a gene. So, suppose this is your wild type protein, which would fold and give you some structure. If there was a loss of function allele, which does not result in this kind of structure, and therefore does not result in the proper folding of the protein, you would it would basically mean that you don't have this protein, and you would get a null mutation, which is also called as an amorph. Uh, when if the mutation that happens in the gene is not it this sorry this going back, this amorphic mutation can also happen if the whole gene is deleted, as you can see here. um yeah. uh, it can also happen that the genetic mutation that happens in the gene does not have an severe effect on the functionality of the polypeptide, in which it can fold itself in a particular fashion, which is not as good as the wild type phenotype, but it's fairly competent enough. Then you But the functionality is slightly reduced, then you get something called as an hypermorph. This might happen if you have a wild type. uh, it might happen that you have one wild type which is fully functioning, and one mutation which is partially functioning, uh, or you might have two mutants, which are both of which are partially functioning, etc. There is also something called as a dominant negative allele, in which the and classical example of it would be that your normal protein is say like this, your uh, mm, your mutant protein is like this. Your mutant protein would somehow sequester your normal protein and not let it function. So basically it interferes with the functionality of the wild type polypeptide. Even if it's present, it doesn't matter. So this is known as a dominant negative mutation, and usually these are hypomorphs. um uh, Usually these result in a hypomorphic condition, in which there is a decrease in the normal function, and it can be called as an antimorph. There are also hypermorphs, in which there is a duplication event, so that your uh, uh, wild type allele has another copy of itself in the genome, and therefore it, instead of producing 10 units, say 10 units of a protein, it can now produce 20. These are called hypermorphs. um yeah. Dominant allele, recessive allele, we have discussed. Monohybrid cross is when you are concerned about a single trait. um So, one point that you should note is that the parent generation in all of Mendel's experiments were purebred lines, which were bred for cell, which were bred for multiple generations, and essentially their uh, genotype was figured out by them. Okay, now. Uh, this is how you would design a Punnett square, that you have your female gametes in one direction, male gametes in another direction, and the cross of that would result in whatever genotype it will have. You have to know the genotype, the phenotype associated with each genotype, to find out the phenotypic ratio that would you would get from a particular genotype, but finding the genotype itself is very easy when you know what the parental genotypes are. One thing that can uh, that exists is something called as a backcross, in which suppose you have you don't know what is the uh, what is the genotype that is associated with your individual is. Suppose it is X, what you can know what the genotype is by essentially crossing it with a recessive individual. And say you have all of them as tall, then it essentially means that this is TT, because the progeny resulted from this, and this would be capital T small t, and therefore always tall. If you are getting some individuals as tall and some individuals as short, you can know that the genotype associated with this is capital T small t. And if you get all of them short, it's small t, unless some organisms are missing out, of course. um, yeah, I think all of these would be discussed later. Does anyone have? Okay, hybrid cross is when you can uh, dihybrid cross is when you consider two traits and you can actually practice all of these. Are there any questions until this point? Are there any questions? Okay. Okay, uh, I can actually discuss the old assignment, and then we can discuss if any of you have any doubts. Okay. These have 10 questions which I thought could be of use. Um, which of the following is not explained by Mendel's law of genetics? We saw that one of his postulates was independent assortment. The second one in which he discussed was basically the fourth one. The second one was dominance and recessive alleles. The third one was segregation. So linkage was something that was found by Morgan and not Mendel. So this is not explained by Mendel's law of genetics. It actually disproves independent law of independent assortment. Uh, okay. Mutant that produces more proteins than the wild type is called a hypermorph because it's more active. Amorph does not is a null mutant. Basically, it does not have any proteins. Hypermorph has low levels of that protein, and antimorph has a form of the protein which sort of does not let the wild type form of the protein to function. If Mendelian genetics, the in Mendelian genetics, if two heterozygous parents are crossed with each other, the phenotypic ratio of uh, the offsprings is 3:1, because as you can um, see, if you have capital T small t crossed with capital T, you draw the Punnett square, you get capital T small t, and you get So this is the result you would get. It's very essential to correctly read the question and see whether the question asks about phenotype or genotype. Because if it were asking about genotype, your answer would have been 1 is to 2 is to 1. But it's asking about phenotype. Uh, but the answer is 3 is to 1, 3 tall and 1 dwarf. If we are the trait we are considering is the tallness of the plant. um, the idea that different pairs of alleles are passed to the offspring independently is the Mendel's principle of independent assortment. um, a plant with heterozygous alleles has blue flowers and one with the one of the alleles codes for white flower. So if it has blue flowers, one of them has to be capital B, and the question says that the other allele is small b, so the plant essentially is heterozygous. And you can, because even though it has a allele for the white flower, it's not expressed, so white is a recessive trait and blue is the dominant trait. So the correct answer would be both A and B. Which of the following statements about enzymes is not true? uh, Enzymes speed up a reaction. True. If it were not the case, you would have to rely on random collisions, and they take a lot of time. That sort of kinetics take a lot of time. Enzyme participate in the cellular metabolic processes. Yes, they are responsible for a lot of processes inside the body. They reduce the activation energy of the reaction, and that's how they work basically. All enzymes are not proteins. If you remember the last week, we had discussed about riboenzyme, which is what basically results in the translation of RNA to protein. And that, even though is an enzyme, it's not a protein enzyme. It's a RNA protein enzyme. Which of the following is the most abundant biomolecule found in the earth? It's carbohydrate, the most abundant. The even though in a cell, proteins are the most abundant in the if you consider as a whole, carbohydrates are the most abundant. A test cross is performed to determine the genotype of a tall plant. Test or back cross. The cross results in equal number of short and tall plants. As I discussed before, somewhere here. Yeah. If you want to determine the genotype of a plant, you cross it recessive uh, with the recessive with a recessive individual. And yeah. So you can guess that since it has equal number of short and tall plants, it should be capital T, small t. In pea plants, seed shape can be round or wrinkled, and the seed color can be yellow or green. Round is dominant over wrinkled, and yellow is dominant over green. In a cross between capital, which is round, green, this is round and green, and this is also round and green. Parents, how many offsprings will have round and yellow seeds? For even one of the offspring to have a yellow seed, you need at least one of the parents to have a capital Y, which they don't. So your answer would be 0. A mutant allele where the protein product binds to wild type protein and inactivates it. It's an antimorph. basically. Uh, One of the suggestions that I have is practice dihybrid process. So if you take Mendel's examples and assign assign a letter to each trait. For example, if you want to study, say, flower color and uh, which is okay, what we say purple and seed. Let's say if you want to study flower color and seed color, so your heterozygous individual will be capital P, small p, and capital Y, small y. So the gametes that result from this can be capital P, small y, uh, small p, capital Y, and small p, small y. Um, basically make a Punnett square, assign uh, these gamete genotypes on both the axis and make dihybrid cross. I urge you to practice this because these would come very handy. Any questions? Does anyone have any questions? Okay. Any questions? I can repeat any concepts if you need me to. Okay, if no one has any questions, I can discuss some other things. So, this is a monohybrid cross. This is if you are considering how the chromosomes would segregate. And so this is the ratios that Mendel found when he did the experiment of the crosses. They not a thing to note is not all of them are 3:1. So this basically shows you the and these are the number of things that he sampled. So this basically is just to make a point that the number of the sampling events are very important. um Coming to genetic inheritance in humans, one of the uh, genetically inherited traits in humans is the uh, whether the earlobe is unattached or attached. And through population studies, it has been associated that capital E is dominant and has unattached earlobes, and small e is attached earlobes. um And if the gene that you are if you are looking at single genes, it's fine, you don't need to check about linkages and stuff. But if you are considering two genes, you need to consider factor in that the two genes might have some linkage between them. And this is essentially the dihybrid cross I was talking about, in which you are uh, looking at two traits and following them. And this is how your Punnett square would look like. In that case, since there were in monohybrid cross case, since there were only two gametes, you got 2 into 2 4. Since here there are 4, you get 16 boxes. If you were to consider a trihybrid cross, which would have 3 genes, it would have 8 different gametes. So the number of gametes that you can get in a cross is 2 to the power the number of genes that you are considering, x. And the square of that basically gives you the size of the Punnett square, if that helps. Yeah, essentially you basically uh, in the F2 generation, you basically cross the this and this results in this, etc. And then you count whatever genotypes are associated, uh, genotypes and their associated phenotypes, and it would give you the genotypic and phenotypic ratio. In a Mendel's dihybrid cross, the genotypic ratio that you would find is always 9 is to 3 is to 3 is to 1. But this is just a phenotypic ratio. This is the phenotypic ratio. The genotypic ratio is a you can just count and write it down. I don't remember it. Uh, I have been talking about the linkage associated with two genes, which was found by T.H. Morgan in his studies on Drosophila, um, in which he saw that when he was following the color of the Drosophila's eye, which can be either red or white, he found that the ratio does not match to what has been proposed by uh, Mendel. And what he found was that there is a linkage between the alleles, and uh, uh, they are not always independently assorted. And therefore, like the alleles are dependent. In this also talked about sex linked inheritance, in which the uh, red colored eye had more probability to show up in females as compared to uh, males. Um, yeah, mutation is the case in which you have suppose you have gene A, and there is a particular genetic sequence associated with it, depending upon and suppose at this position there should have been an A, but instead it mutates during either replication because of some genetic error, because the polymerase that you have is not error it's not error free. It's not very prone to error, but it has an error. Or due to some uh, mutative agents, it became G, say. Um, then this particular uh, base would change itself for every mutation. So you So your gene is basically not the same. Depending upon how big your mutation is, there are different kinds of mutations, such as point mutations, in which only one of the bases is changed. Frame shift mutation, in which, say, you have this sequence uh, of nucleotides, you have something inserted. Suppose one base that is inserted here, it basically shifts the sequence that is downstream of it, such that the when the when transcription occurs and finally translation happens, the codon triplet that the ribosome would read is not the same. There are also inversion mutations in which you take this stretch of DNA and then invert it, and there are deletion events in which a stretch of DNA is deleted from the genome, etc. Nothing as bad as X-Men though. Uh, yeah, that's about it. Does one have any questions? I can explain any other concepts that you would like to understand as well. Does anyone have any questions? Otherwise, we can end the session. Are there any concepts that you would like me to explain again?
Hi friends,
Sorry? We can end the session then. Thank you, everyone. I am going to end the session. I don't think there are any more questions. Thank you.